This is Unit 6.3. Gene expression and regulation. Why does gene expression have to be controlled? Remember that all the cells in the body come from one cell, the egg being fertilized by the sperm, and everything except egg, sperm, and red blood cells have the same exact DNA because mitosis occurs, and you have different cells that produce different things and act differently because of cellular differentiation. Remember, that's when cells only read certain recipes in the DNA. So only certain genes are expressed in a particular cell, even though each cell contains the instructions to make every protein that that organism can make. So, gene expression, also known as a central dogma or protein synthesis, is turning the DNA recipe into that protein, and that's that two-step process of transcription and translation. So, both prokaryotes, which are bacteria, they do not have any membrane-bound organelles, and eukaryotes, which are multicellular with membrane-bound organelles, have to control the types and amounts of proteins, but they use very different methods. Remember that prokaryotes do not have a nucleus. Their translation can start as soon as transcription has begun and there's an mRNA being produced. So prokaryotes use operons to turn genes on and off. It's like a light switch. Um, Transcription and translation occur at the same time in bacteria because there is no nucleus. And this is what we looked at prior. This is a eukaryotic gene where you have your promoter Tata box, and then you have your RNA polymerase that comes and transcribes that particular gene. Where does translation and transcription occur in prokaryotes? And that is going to be in the cytoplasm. Where does translation and transcription occur in plants? And that is going to be B, in the nucleus for transcription and the cytoplasm for translation. All right, so that gene regulation in prokaryotes is done by an operon, which is a portion of DNA that contains the promoter. This is where the RNA polymerase lands, and we've seen that in our virtual lab. The operator, which is the section of DNA that acts like the light switch, it allows or prohibits transcription. And then the gene, which is the structural recipe for the protein. So here we see our operon with our promoter region, the operator, and then our structural genes. This regulatory gene is usually pretty far upstream and codes for the repressor protein that will land on the operator to prevent transcription of that gene or series of genes. So the lac operon is a well-known bacterial gene regulation system. It's very well studied. And lac refers to lactose, and which is the milk sugar, and lactase, which is the enzyme that can break down that lactose sugar. So when lactose is present, it binds to the repressor protein, which blocks the operator. So it prevents RNA polymerase from transcribing the downstream genes. So this is a video. I suggest that you watch it. It shows the structural changes when the repressor protein is exposed to lactose. Which of these is not part of an operon? And that's the regulator, is not part of the operon. So the repressor protein loosens its hold on the DNA at the operator when lactose is present, because lactase should be produced in order to break down that lactose so that that bacteria could eat. When there is no lactose, it would not benefit that bacteria to produce an enzyme to digest something that's not present. So this is conservation of energy and resources by not producing enzymes that are not useful for that organism in that environment. So being able to turn this on and off depending on whether lactose is in the environment or not is helpful to those organisms. Now, that was prokaryote regulation. They use operons. Eukaryotes do not use operons. Instead, eukaryotic gene regulation occurs at either pre-transcriptional, before the mRNA is made, transcriptional as the mRNA is being made, post-transcriptional after the mRNA is made but before it leaves the nucleus, and translational. Which of the following do eukaryotes not use for gene regulation? And that is an operon. So eukaryotes do not use operons for gene regulation. Which of the following do prokaryotes use for gene regulation? Remember, prokaryotes are bacteria, unicellular organisms that do not have membrane-bound organelles, and that is an operon. Right. Now, this again is for eukaryotes and the 4 different places where gene regulation occurs. Pre-transcriptional involves the epigenome. So the epigenome are the proteins associated with DNA regulation. So remember, we talked about those histones, which are the protein balls that DNA wraps around, and certain methylation or acetylation of the DNA can cause certain parts of the DNA to be wrapped around these histone balls. So when the DNA is coiled around a histone, it's not able to be transcribed because it's wound around something, and RNA polymerase cannot go to that section of the DNA and transcribe that gene. So it can be made accessible by being between the histones, where it can be readily accessed by RNA polymerase, or it can be on the histone and wrapped tightly around there so that it can't be accessed by the RNA polymerase. And this is something that can be done in response to the environment. Unlike changing the nucleotide sequence of the genome, which is a mutation and is very rarely good for the organism, you can change what genes are being transcribed in an organism in response to the environment. So this pre-transcriptional regulation of the genome is a great way to respond to environmental conditions and maximize those genes that are optimal for that environment. So epigenetics is actually the study of the factors that help regulate gene expression. And these include environmental factors, what the organism does. So, a proper diet, lack of sleep, stress, exercise, all of these impact the epigenome. Pathogens also impact the epigenome, and your microflora. So the microorganisms that live in and on you also change your epigenome. So we've talked about this in class. They are even doing fecal transplants with gut microbes that change a person's epigenome based on just the microorganisms that are living in their intestines. And then you can have cell signals from other areas of the body, so hormones, electrical signals, or even cell-to-cell contact. So it was recently discovered that epigenomes are, in fact, inherited. Previously, they thought that when egg and sperm came together on date night, that the DNA was wiped clean. All the proteins were kind of thrown out and it started fresh. It turns out that some of your parents' epigenomic markers are inherited by the offspring. So this actually sets up the organism for being optimal for the environment because the parents are living in that environment already. And remember, you can change your epigenome anyway. Epigenomes are the proteins that are involved in the regulation of gene expression based on environmental factors. Remember, it's a way to control the genome without impacting the DNA sequence, because that is not a mechanism that's viable. All right, so that was pre-transcriptional, transcriptional regulation. Remember, each gene is controlled by that promoter. And if you remember the virtual lab where you looked at the positive and negative transcription factors that helped RNA polymerase start transcribing a particular gene. So really this transcriptional regulation is looking at the enhancement or silencing of transcription by RNA polymerase. You can also have post-transcriptional regulation. Obviously, this occurs after the mRNA has been produced, but it happens while it is still in the nucleus. This includes your cap and your tail, and it also includes removing those introns, which are non-coding portions. And don't forget that when you produce an mRNA, you can leave the introns in, you can take the introns out, so you can produce multiple proteins from this same gene, depending on what is going on in the cell. So it gives a lot of variability, again, when you don't have the ability to change the DNA sequence, but you can change how that sequence is expressed in that organism. Then you have RNAi, which is RNA interference. This came when scientists were working with microbes and working on genetic engineering, where they were using sections of DNA from other organisms to try to get that to be expressed in bacteria. So it turns out when DNA that is not native to that organism is put into a microbe, you have RNA that interferes with the expression of that DNA. This is considered post-transcriptional regulation. And then finally, you can have translational regulation. So we didn't really talk about this, but when you have a single mRNA molecule, you can have multiple ribosomes reading it at the same time. So they'll be on all different sections, but you don't have to wait for a ribosome to completely read that mRNA before another one starts translating it. So you can have this happening at multiple places on the mRNA at the same time. So you can control the number of ribosomes that are being devoted to each mRNA. You can take them, they can have a different job, or they can be concentrated on a particular mRNA molecule, and that will increase the translation. So you'll increase the amount of that protein that's being produced. All right, removing introns would be an example of, and that will be post-transcriptional, right? Because the mRNA is already made, but it's still in the nucleus. Methylation of DNA would be an example of, to rephrase this, when you are making certain genes accessible or inaccessible via the methylation in histone proteins, what would that be an example of? And that is pre-transcriptional. And the dead giveaway is when you see DNA. Obviously, it doesn't have to do with mRNA. RNAi is an example of, and that's post-transcriptional regulation. Having more ribosomes available for a particular piece of mRNA would be an example of, and that's translational regulation. Having negative transcription factors preventing binding of RNA polymerase is an example of, that's transcriptional regulation. Right? So you're either encouraging or preventing transcription by RNA polymerase. All right, so what are the factors that influence these regulations of the genes? You can have both internal and external factors. Internal includes signaling between cells, contact with other cells. Remember those HOX or homeobox genes that allow correct body plan to be set up. That relies on cell to cell contact, but you can also have hormonal expression during differentiation. Even exercising in humans has been shown to have changes to target tissue DNA. Diet, we've talked about the microbes in your intestines. So all of these are considered internal because they're within the body. And then you can have external factors such as light and temperature. So some organisms use light via pineal gland to set their circadian rhythm or even to change the fur color. So you may have seen rabbits or fox, caribou, where they'll be brown during the year, and then during the winter they'll be white to blend in with the environment. So the deposition of that melanin, which is the protein that causes coloring, is changed so that that gene expression is changed by external factors, usually amount of light. And then you could have drugs and chemicals, even nutrition. Those are all considered external. They can have influence on a developing fetus, tremendous influence. And we talked about how your parents exercising or what drugs they're exposed to can also impact your epigenome. And summarize the difference between eukaryotic and prokaryotic gene regulation. So, prokaryotic gene regulation occurs via operons. Eukaryotic gene regulation is more nuanced and includes pre-transcriptional, transcriptional, transcriptional, and translation regulations.